welcome back. Thank you for being here today in the afternoon. Today, we're going to try to finish up with SIR modeling and continue with some variations of SIR modeling. And the thing that I think probably is most important is that we're going to try to do a little bit of fitting of model results to experimental data. We're not going to go into huge detail on that today, but we'll get started. And then we'll come back to it and go into more details about how fitting works in subsequent lectures. So the first thing I had was actually you, which is I wanted to circle around the room, have people screen share and present some of their ideas about possible projects. And again, I know that it's hard to do that when you haven't had a lot of class, but I hope that people have some ideas and would like to present. So is there somebody in order that we would like to go in, or should I just go around the room based on who's in order and on Zoom? Maybe start at the end. Mercedes?
Hello.
Hello. So let me unshare, and you can now should be able to screen share and let us see what you're thinking about.
Yes. So right now I have been looking into either modeling cancer or modeling brain neural activity. However, I'm running into the issue of not having access to a lot of the articles without paying for them when I try and read them. So let me finish sharing here. The two papers that I've been looking at for the, let me present here. For cancer, the top one was going into a little bit more about the mathematical modeling of the cancer cell and comparing it to a treatment. And then the second one was going into the tumor cells developing with your immune system being compromised by your diet, and just kind of how those two compared to each other, and how the tumor cells modeled. I also ended up reading a paper that Professor Macklin wrote, which was pretty funny that I just ran into that while looking into this. I didn't write it down there. Oh, can you still hear me?
Mm-hmm.
Okay, sorry. I thought the internet or my headphones had changed. And then the one that I found for the neural activity was going into just how you can track down where individual neurons are coming from. I didn't really like the paper, but it caught my attention with the modeling they were using. So that's kind of what I have gotten through so far.
So in terms of access, if you set up a VPN client to the university, or you log into the university libraries using your IU password, you should be able to get access to papers.
Right.
Okay.
I posted the link in the chat. You can access Google Scholar through the IU catalog. And then once you look for papers, if the university has a license with the paper, with the journal, you should have access to the paper, and they do with most of them. But you have to look it up through the link, through access through the IU catalog.
Yeah.
So we should probably try to meet. Tomorrow is probably going to be bad, but either Thursday, Friday, or over the weekend, and look at the papers together. The main thing that I want to be careful about, if it says an ODE-based model for tumor cells and immune cell competition, that sounds good.
Yeah.
I want to make sure that there aren't things hiding in the paper that make it difficult.
Okay.
So for example, if they're delay equations or it requires integral differential equations, those are things that are more complicated to do computationally. And so I want to try to make sure that people don't accidentally pick a paper that looks easy and actually has something hiding in it. that's hard. But those are all good. They're all very good topics, and people have done quite a bit of modeling of immune response to cancer over the years in this class, and there are plenty of good papers if what you want to focus on is immune response to cancer or cancer therapies or PBPK models of cancer treatment. Those are all quite possible.
Yeah, I think
So,
that's what I wanted to focus on. So
okay, that's fine. So, but let's try to talk in the next day. Again, tomorrow's not going to be easy, but by let's say by next class, let's have had a meeting
Okay.
and go over that. I don't know if anybody else in the room is interested in cancer modeling. If they are, it's fine to team up. Again, people are welcome to work on their own. They're welcome to work in pairs, either way. Of course, if you work in a pair, I expect you to do a little more work so that the amount of work per person stays more or less the same, but sometimes it's interesting to work as a team. Does anybody have any comments or suggestions? I know we haven't talked about cancer modeling in this class so far, but that's fine.
So would the goal of this cancer project like this be to replicate the model present in the paper?
Well, I hope that people can start out by replicating. I mean, so the base assignment is to replicate a model that's present in a paper. Sometimes people are a little more ambitious and they want to take something that hasn't been modeled and model it from scratch. That can be challenging, but people have done it. But if you take a model in a paper, after you've replicated it, the aim of the paper of the project would be to extend it in some way. For example, you might have, say, a model of tumor cells and immune cell competition, and you might read another paper about chemotherapies. And so you might add to your base model of tumor cells and immune cells a model where you hit the tumor with a chemotherapy where part kills part of the cells, and then you see how it responds. So the goal would be that you should, if you take an existing paper and replicate it, you should also try to extend it in some way.
Okay, thank you.
It depends a bit on how elaborate the model is. So if the base paper takes a lot of effort to replicate, it's very complicated, then maybe extending it isn't so reasonable. If the paper is something that you could replicate in a few hours, then combining it with another paper or extending it in some way is reasonable. But that's one reason to start early on it, so that we can talk about those things and come up with a reasonable plan. I guess you're the next on the line. So what are you thinking about, Kali?
All right, let me share my screen. So the main idea that I had was to try to explore a vaccine model for the COVID-19 pandemic. So basically, we've all been hearing in the news, finding a vaccine for COVID is kind of seen as this holy grail to stop the pandemic, but it's kind of hard to tell what will actually happen when we have one. So this project idea would aim to explore models predicting the impact of a vaccine on the COVID-19 pandemic in the US. I figured we might as well just focus on only one country because everyone has different trajectories. And obviously, for some preliminary hypotheses, we would expect the number of infected individuals to decrease with a greater vaccine efficacy, a greater number of vaccinated people, and a greater vaccination rate. Okay, so the necessary information to create this model, we would need to have the R0 value for COVID-19 and if possible, an estimation on infection probability, the latency period for COVID, the illness duration for COVID, and statistics on the number of cases and number of deaths in the US, possibly separated by geographic region as well. It could be worthwhile to potentially look at vaccine impact in different areas. Say like a densely populated place like New York could have a different model than like a rural area. And if possible, it would also be nice to have some estimates on asymptomatic cases, but a little bit more on that later. So then there are potential models to explore that kind of vary in complexity. So there's SVIR, so susceptible, vaccinated, infected, recovered, SVEIR, which includes asymptomatic or exposed cases. And because there seems to be some evidence that people can get reinfected with COVID, that's where the SVEIRS model comes in. So there are a lot of potential opportunities to explore here. I've compiled a few information sources. I think a couple of these were off the paper reviews in the Google Drive and a couple I found myself. So there are 2 papers about SVIR models, one of which is more complex than the other. The more complex model includes statistics like recruitment into the population, which I think probably means birth rate, and then like natural death rate. I don't know if that's necessary to include, but I thought it'd be interesting to at least consider both of these papers. We also have an SVEIR model for an imperfect SARS vaccine back from 2003. COVID is in the SARS family, so maybe there's some potential there to do some looking. There's also a paper I found called Modeling the Role of Asymptomatics in Infection Spread, and it's specifically applied to COVID-19. And it doesn't include vaccination, but I think that because it specifically looks at asymptomatic cases, that could be used to help inform the model regardless. Then obviously to get the pandemic statistics, CDC has a lot of information, and also the Johns Hopkins Resource Center that we looked at last week. So then for the NanoHub application, I'm kind of envisioning this as more of an exploratory educational tool that would allow users to select a model type, change parameters, and see what happens. This would ideally also be a GUI, but we'll see what I can do by December 1st. And that was my main idea that I had. I also looked at some other non-epidemiological project ideas. This is the one I was most excited about. At one point, I had an idea to try to model COVID in a university setting using IU testing data, but I figured by the time we all go home and we have all the mitigation testing data, and it's November, it's probably not enough time to really build the model by December 1st. So yeah, that's it.
That sounds fine. I think it would be great to have we're going to do some of these basic types of models and a little bit of fitting in class, but one can go a lot further with these things than we're going to be able to do. And I think having a demonstration like that, actually, making the GUIs in Jupyter Notebooks, as Josh will show, isn't so bad. You have to learn how to create sliders in Jupyter Notebooks so you can change parameters in a GUI way. But it's really not too bad. Okay. It's only a few lines of code. So I think that's a reasonable thing to do. And there certainly are plenty of classes of models. And maybe especially thinking about compartmental model, where you have your example of city versus rural, where you have different time scales for the infection. That could be pretty interesting. I know there's been some worry about how an imperfect vaccine might actually increase rather than decrease infection in certain cases. Some sort of paradoxes there. I think we can definitely build a project around this. I think that you thought it through pretty well. So again, we should try to meet in the next few days, probably. I'm less concerned here because it looks like you're pretty far along in terms of what you're thinking about. I'm more urgent to meet with the people who don't have a project to help them get started. But I think that's great. And again, if people have shared interests, it's fine to team up. There's certainly people who've done, the Perlson papers, trying to look at essentially what happens if you have imperfect control within an individual. And those have also their own interesting questions. But I think given the urgency of a vaccine and the questions that come up, that could be a great problem. I don't know if there any work or ideas about one of the things that's been particularly tragic about COVID has been health equity issues. So minority and poor communities have had much higher infection rates, and the people who are infected die with much higher rates. And so there's potentially a question about access to vaccination and issues in that regard as well that could be explored, perhaps. So I think it's a great problem.
Okay, thank you.
Okay, thank you. Okay. Jeffrey, have you made any progress on your thinking about maybe a neural model? I can unshare you if you're.
Yeah, I can figure out how to share this. Hold on. Is it sharing now? Yeah.
Mm-hmm.
Okay. Okay, I don't know why it's doing this. I have so some just basic backgrounds on neuroscience. These books, the first one I'll probably work all the way through, but these last two, I need to email him about that one still, the third book.
We just had a phone call with him today. 2 o'clock every Tuesday.
I need, but those books will probably just be a few chapters out of them. And so I decided to look more into bursting and synchronization. And I have this first paper here, kind of has a model of interacting cortical columns. And this second one kind of just goes into the theory and what cortical columns are, because I wasn't so familiar with that. But there's a kind of interesting model in this paper that I'd like to look into. And I have these other papers by Dr. Ermintrout that I've kind of just skimmed through so far. I haven't fully read into them, but they sounded interesting. And I kind of want to see what this inhibition stuff has to do with bursting. So I'm kind of still in the progress of reading about bursting and synchronization and that. But I think overall, I would like to take one of these bursting models and try to apply it to an icing model from like thermodynamics, which is a little different from what we previously talked about with the lattice Boltzmann. But I think it is maybe more appropriate because I don't think it depends so much on the spatial representation that you mentioned with the lattice Boltzmann, or the spatial kind of dependence. So I'm looking into this, and I've seen that the icing model has been applied to neural systems and neural networks before. So some of these papers kind of go into that in a little more detail. I've only read this first one so far and just kind of skimmed through the other two. So that is pretty much my goal for the project at this point in time, is to take a bursting model and apply it to an icing model and see what kind of information you can get in that. And then this is not so important. That was just lattice Boltzmann stuff for other maybe longer term ideas. So yeah, that's where I'm at right now.
Right. Again, I mean, I guess it's not absolutely essential that we write something that can be expressed as a coupled OD model for a project. the class will do, I mean, everything we're going to do in the class will be coupled ODEs. And so if you go outside of it, like the Lattice-Boltzmann model, there's nothing wrong with that. It's just not going to be material we're going to be able to cover in class. It wouldn't be bad for people to hear about it in your lecture at the end of the semester. But those are fine. Again, if you're interested in the relationship between Ising model and neural networks, Javier, a postdoc in my group, his background is in solid state physics, and he's done a lot of work on magnetic systems, including Ising model. And he's working now in neural network, not biological type neural networks, but rather computational deep learning type neural networks. And so I could definitely connect you with him. He's tried to do some simulations using TensorFlow, sort of standard off-the-shelf neural network, to try to simulate Ising model behavior. So far without enormous success. So, but he might have some ideas about that. So let's follow up next. Why don't you do a little more reading over the weekend? Let's follow up next week. Look at some papers that you've picked out and walk through them and see what we can pull together.
Okay. Yeah. Thank you.
But I'm happy to help out.
Okay.
Okay, great. Okay. Let's see who's next on our list. Devin.
Yeah, can you hear me? Okay. All right. So I wasn't too sure what I wanted to do coming into the project. So I started kind of surfing through different papers, and one of them that I came across was oncolytic virus dynamics. And so oncology, obviously, it's a study of cancer. And so this method uses viruses that infect and kill cancer cells and leave regular cells healthy. I found that interesting just because we usually think of viruses as bad, but this helps medically. These viruses can be found naturally or modified in a lab to be suited towards the type of virus or type of cell you're trying to kill. And so a lot of people are trying to use them as a form of cancer therapy to spread through the tumors and kill them while not disrupting the normal cells. And these aren't too far developed. A lot of them are still in research and clinical trials. I did see one that was FDA approved. I don't quite remember what it was. I think it was to kill like melanoma or something like that. And so for my model, the inputs and outputs that I would use are the cancer cell growth and death rates, and then the virus spread rate. Like if the virus spread faster, kill cancer cells faster. And then I'd also use the total cell population and the numbers of infected, healthy, and removed cells. And then for the output, I wasn't entirely sure what the output would look like, but ultimately you just want to see whether the virus is managing to spread far enough to kill the tumor that you're going after. And so one of the sources that I used was pretty well developed. It uses OD modeling that is dependent on the rate of the virus spread and also whether the tumor was bounded. So whether it could, whether it was at a stopping point or whether the tumor was continuing to spread. And then it looks at whether the equilibrium is achieved. So there's no more tumor growth, the virus is throughout it. And then some of the problems that you might run into with this is of like tumors. There's not a lot of like fundamental mathematical laws that kind of define these tumor spreads. So a lot of the stuff that they're using now is qualitative data. So just looking at how it spreads and some empirical data. So measurements you can see. And then also most of the models right now are pretty generalized. So there's going to be a wide difference between different cancer cells and the viruses that you have to use to target those sort of cancer cells. I was There are some models that are out that are suited to one type of cancer cell and one type of virus, but I was probably just going to go with one of the general types. And then for kind of ways to expand, I could look into sort of a cancer virus specific model for a certain type of cancer. And then also just getting more specific mathematical laws.
So that paper, Natalia's, the Komarova paper, so that would be, so that's already on the subject. So one, you could start with that and replicate that to begin with, presumably. So we should sit down and look at that one together. Josh has done some modeling of cancer. Not this particular kind. I don't know, Josh, when you were out at UC Irvine, did you meet Natalia? Was she one of the teachers in that course?
I don't.
Because she's on the faculty in the Mathematical Biology Institute at UC Irvine. Vodart's a big name also. So I feel some confidence when I see a paper with their names on it that it'll be interesting. They're both very, very highly respected. They tend to be quite mathematical. So as you pointed out in your discussion, they tend to write relatively abstract models, rather generic ones, which is fine from where they come from. But I think that could be quite an interesting problem. Use of viruses as control strategies is something that has been used, actually is an old method, both for cancer and for bacterial infections. People used phages, which are bacteria that infect, viruses that infect bacteria, before they invented antibiotics. And then when antibiotics came out, they tended to displace the use of phage, although in the former Soviet Union, they kept using phage. And there were some attempts to use viral treatments and bacterial treatments for cancer before radiation and chemotherapies were developed. And it's always interesting when these kinds of things come sort of first full circle and they come back to ideas that were developed 100 years ago or more, and are reinvestigated. I think that's a great problem. So it's interesting. I mean, as I say, we picked the example of viral infection for the theme of the examples in the class, but the methods are all quite general, and so using them in the context of neuroscience or cancer, I think, is great. It'd be good to, since we're doing the viral examples in class, having you do non-viral examples and show what these methods are capable of is particularly helpful. I think that's great. Again, we should meet and try to discuss details a little bit more. Okay, let's see, Brian. So far, I haven't seen any obvious pairing up, although you're certainly free to discuss that offline or together if there are specific things that you think would be good.
Okay. So I as well was sort of just thinking about the problem. Let me see if I can share the screen. And I didn't have a lot. I didn't actually come to any conclusions. Also, these slides are not very well made, but so I came to a couple of ideas. I think the modeling, the type of modeling, or the thing that I would like to model, the problem, would have to concern COVID-19 because it's just such a present topic and it's so widely affected our lives. But I'm not really sure where to go with it. So I kind of thought of a couple of ideas, such as the first thing on this slide deck is, age-dependent severity of COVID-19 cases. So we know that COVID-19 affects different people differently in the form of age and I guess I think it was like discrepancies in wealth and stuff. I wanted to see if I could model that. Another thing that I wanted to see is if this could probably be like really hard to do, but I wanted to see if you could model population quarantining and seeing how like a percentage of people quarantining would affect the spread of an epidemic. Also, herd immunity was something I wanted to take a look at because I wanted to see like what would the like amounts of people, what percentage of people do we need in order to have a situation where we can have COVID-19 herd immunity? Something interesting. I also came across one of the same things as Cali, where I wanted to see like how vaccination would affect our world if one was released currently, because there have been talks specifically like surrounding the presidency and like politics. It's just like if a vaccination was produced right now and the efficacy of it was evaluated, how would that affect our current situation right now, right? With however so many people that we have currently that are currently infected, and how like the manufacturing of that, how like the rate that we can get it to other people. And then the other one that I thought of was modeling the differences in person-to-person interactions. So like if someone went to a party or if someone decided to stay in quarantine, what would that look like?
So those are all really good problems. Again, it's a little bit like some of the things Jeffrey's interested in, where some of those are easier to do with the specific methods we're covering in this class. Some of them are certainly achievable, but require using some methodologies that are not things that are going to be a focus of this semester. There's nothing, again, I'm not going to tell you you can't do something that requires additional methodologies. But for example, the chain of transmission models, the stochastic chain of transmission models that people like Vespignani do, and there are a lot of very good ones of these. Those are very interesting, but they require somewhat different computational infrastructure from what we've been developing. And so it would require a little bit more coding, independent coding on your part. One can certainly do it in Python, but you're sort of on your own. In other words, we wouldn't have the equivalent of antimony to help you out. One thing to think about, depending on what other people have done, is to look at some of these models like the ones nobody so far looked at as proposed doing a model. A lot of people picked up the Goyal paper as an example in their paper lists, but a paper of actually how the body responds, a single individual responds to the virus, and how the virus spreads within the body. As we'll see either this week or next week or the week after, it's very similar to what happens in terms of infection in the population mathematically. But those are some things that we could think about. We'll talk a little bit about herd immunity today. In fact, that's one of the first topics we'll get to today in class. Mm-hmm. And so I think these issues are quite interesting. It would be good to, and the other thing to think about is some of these papers, which some people mentioned in their lists, about how to unlock. Is how do you open up again after you've done a quarantine safely. And there are interesting issues with vaccination. I think I mentioned there are vaccines that have existed for a long time. I have been vaccinated for plague. Of course, that's not viral; it's bacterial. But nevertheless, I've been vaccinated for plague. Most American doctors don't even know there is such a thing as a plague vaccine. It's actually been around for 70 years. There's a vaccination for tuberculosis and the issue with both of those vaccines is their efficacy isn't very good. And so there's a lot of argument then about whether, for example, the risk of the vaccine is worthwhile. There are other issues that come up when you have low efficacy. In general, as we'll see today when we start talking about R0 in more detail and herd immunity, anything that reduces the transmissivity of the virus is going to reduce the number of people who get sick. And so in that sense, even an imperfect vaccine is probably a good thing. But there are a lot of caveats. Suppose you have a vaccine that kills 1 in a million people who get it. That doesn't sound like a lot, but with 1 billion people, that's 1,000 deaths. If the virus wasn't going to kill very many people, then you might have a problem. And so that tends to be an issue that comes up politically, especially when a disease has been dealt with very effectively, like polio. Then the cost-benefit analysis becomes very complicated. So I think there are a lot of interesting issues you bring up, and I'd like to, again, to talk to you. It sounds like you and Kali are thinking along somewhat similar lines. So maybe the two of you could talk together and see if there's some sharing of ideas. Anybody else? Josh, do you have any comments on that or suggestions?
I do not know. It's gonna be interesting to look at what other people have done. And
right. I think the thing to do would be to start out with studies with models of vaccination efficacy for other diseases, and look at what mathematical modeling has been done of that. Because there won't be a lot there'll be some basic things probably for SARS, but I'm sure, for example, influenza is a classic example because influenza is a disease that's been around forever. The vaccine's been around for quite a while. The vaccine is relatively ineffective. And so it's quite interesting. And I'm sure I know there's been a lot of mathematical modeling. There's a lot of epidemiological data. One of the things about influenza that makes it a little more complicated to analyze is it has a very complicated spatial structure. So influenza epidemics jump northern hemisphere to southern hemisphere twice a year. And they almost always start in one big city in the southern part of the United States, for example. In the US, the influenza epidemic almost always starts with a single infection in a place like Houston or Tampa, and then spreads and jumps city to city. You can look at the spatial distribution of it. Every now and then it does something strange, like starting in Rochester, New York. But those kind of models, those kind of spatial models, are a little harder to do in the context that we're going to have in class. Although you can do compartmental models where you sort of have simulated 10 cities and the exchange of virus between the cities. That's possible to do. So those are things to think about a bit.
Yeah.
Tyler?
Oh, can you hear me? Let me pull up my screen really fast. Okay, so I'm primarily interested in the within host kinetics of viral infections. I was between this and cancer, but I think this might be the way I want to go. Some potential models I've read over in the papers are mostly SIR models. And in one of the papers I was reading, there was an extended SIR model that I read into. Mostly interested in this because the immune response to the flu virus of all sorts is mostly unknown still. And there's a lot of speculation as to why there's a flattening of the virus during an infected person's interval of infection, I suppose. And sometimes there's a second spike in the amount of virus. And it's that speculation that kind of drove me to this topic. Some of the inputs I thought we would need, the initial amounts of cells, free virus, amount of infected cells along the way, and as well as the rate of virus production and the infectivity and the probability of one cell infecting another and another. And like I mentioned before, the immune response and the rate at which the immune system can clear the virus and natural clearance of the virus and how that affects the infectivity. The output, I am somewhat uncertain, apart from the amount of cells that are susceptible, recovered, or infected. And that, yeah, I'm not really sure how the output's supposed to look apart from this. But some application and practical uses I thought of, like Callie had an educational tool primarily. I thought something along those lines of changing parameter values for different viruses and seeing how the outcome might play out.
Right. Well, Amber Smith has that nice review on different viral kinetics, and she has those couple of models which are very simple, and then there are all the multi-compartment models of Burleson, which are also pretty simple. Simple doesn't mean bad, by the way. And then Bard's old student Erica has some very elaborate ODE models of influenza response, maybe too elaborate, which we've been talking about. So definitely that's an area where Josh has been doing simulations of this very specifically. We may or may not have some fancy new models available to us, because Bard Ermintrout and Erica Mohan have been working over the summer and now at the beginning of the semester with a group of students at Pitt, developing a new model of COVID infection, OD model of COVID infection. And they're not willing to share it until it's published. So, but if they get written up, maybe we could play with it. It sounds quite promising. But I think this is definitely something one could do. And I think it would be good for at least one. We'll do it. I mean, we'll do some of this in the next couple of lectures. In other words, if I look at the slides that are sitting at the end of my deck that we haven't gotten to yet, they're a model of precisely this type that Josh developed. But it's not fit in detail to any specific virus, although it's basically mimicking influenza dynamics. And so yes, so I think that's a good choice. So pulling some good examples would be the place to start with that. Find some papers that you like that are useful. Again, there's certainly going to be some for COVID, but I'd start looking at models for other viruses and see what's been done. The immune response, the models of the immune response can get arbitrarily complicated because the immune response can be arbitrarily complicated. But as Amber showed in her flu models, you don't need very complicated models to be able to replicate a lot of features of the viral load curve. So there's plenty to do there. That will definitely clarify over the next week or two as I introduce some of those ideas. So let's try to meet sooner rather than later to talk about some of those things. But Josh has done a lot of reading on the topic. It can help us out on that. Okay.
Okay, thank you.
Great. Antonio, you have I think you're the last on the.
I am. I am. Admittedly, I saw a NanoHub application on the project parameters, and I got a little carried away with that specifically. So I'm a little worried that I need to go back and kind of redirect and specify a little bit more on my idea. But here's what I have so far. I'll just run with it, see what you think. So my initial idea was more of a tool to kind of roughly model a novel virus spread. So the general idea would be that in the case of a novel virus, researchers can pull what they know about the virus early on, plug them into this model, and get some outputs in some nice graphs. And as they learn more about the model or the virus, they can update that model and continuously improve the outputs and the predictions. So I think initially I'd just start with an SIR type model just to get the rough estimate of the initial spread. And then I just kind of started thinking of some iterations that you could add to this kind of tool, allowing researchers to choose how many population centers they wanted to include, and then being able to categorize them as large, medium, and small, or allowing them to specify kind of the flow of people between those centers. Also, the ability of researchers to test a vaccine. They could input the effectiveness in reduction of chance of infection, the duration of effectiveness, as well as the percentage of initial population that is vaccinated. And I think also that they would be able to kind of play with this tool and realize that, if we want to drop the infection rate by X amount, how effective does the vaccine have to be? What's the benchmark? So they can kind of go backwards and look at that as well. Another iteration could be allowing researchers to plug in like different health initiatives and see how they affect the rate of infection, and allowing them to input hospitalization and death rates. And maybe when they input these values, which would be optional, it could kind of further refine the model that's being used. So instead of an SIR model, it might evolve into an SIHR model or an SIRD model to kind of input those. And all of these kind of states, I think, in my head would be kind of drop down, and they'd have default values, but the researchers would always have the option to input very specific values for those as well, such as population density for the size of cities and whatnot. Some of the inputs and outputs, base inputs, infectivity, the working baseline with their R naught values, what they have at the time, kind of a choice of the behavior of the virus, whether it's seasonal or not. Total population currently infected, total population that they want to study, the currently infected population, and the range of time. And they'd put all those into some GUI and it would spit out the graphs detailing the spread and the values of the parameters that it used. And I think it'd be really interesting to kind of use this tool, again, just so researchers can really roughly, quickly kind of input what values they know and see what pops up, see how it's looking, see how it's improving. And if they just wanted to theoretically test predictions and test scenarios that they could exemplify those predictions to others and say, here's what happens when you mask, here's what happens when you do all this, and here's what happens if we had a vaccine of X effectiveness, whatnot. Here are some possible resources that I've found. This first paper details the rest. I'm not even going to try to pronounce it. I'm a little too tired for that right now, but basically what I pulled out of this paper was that they included a vaccine model. So they had kind of how effective, the duration of the effectiveness of the vaccine, and what that kind of did to the curves. They also included a seasonal model as well. So it was kind of interesting to see them integrate like the cosine function into their model. The second paper, it has just really good descriptions of a lot of the ODEs that could be implemented into this tool. And they're very clear in the parameters as well. And this last paper I read, it was a paper detailing the spread of the virus in Singapore with different initiatives enacted public health wise. And so it would be a good paper to draw on to kind of modeling those different public health initiatives.
So I was just going to I'll share and send an email. My old student Yi, who's teaching mathematical biology this semester, was assembling a list of epidemiological models of COVID, and I will forward it to everybody in the class. I was planning on doing that before, and I didn't do it. So the title of the eval may not be that descriptive, but if you look for it now, it should be. So there are a couple of people that I think that's great. I think there are a couple of people that are interested in trying to put together a tool for exploring epidemiological models. And so maybe two of you could pair up and work. Again, you don't have to. Sometimes it's interesting to have two people work on the same problem and see how different the results are. But if you like, you could do that. Your point about seasonality is well taken. It's not something we're going to cover probably in our little epidemiology section of this class. But if the basically R naught changes seasonally, then you could have interesting effects. If the time for the virus, if the time for the epidemic is short compared to the seasons, then it doesn't do much. But if the time for the epidemic is long compared to the seasons, then it does do quite a bit. As I mentioned, flu is the classic example where it jumps from the northern hemisphere to the southern hemisphere, then comes back repeatedly. And so I think the idea of then putting that in a framework that people could use. There are some modeling tools of this kind that exist, not exactly the one you've described, in other frameworks. So, and making something like that available in NanoHub, I think would be great. So I think it's a good project. Again, the trick is always to try to narrow it down to something that's achievable. But let's again start with the papers that you've identified as interesting, and the reason I tend to suggest that people start by replicating a paper is that gives you something that you know you could do. If they've already done it, you know it's possible. And then if if they've already got results, you can check, you know that there's a correct answer, or at least if they haven't messed it up. I remember once or twice during the course of these classes, a student has picked a paper to replicate, then discovered that there were mistakes, fundamental mistakes in the paper they were replicating, and actually corrected those mistakes. So just because it's a published paper doesn't necessarily mean it's right, but it gives you at least, usually it's pretty much right. And so that gives you something as a baseline. Whereas if you start out a project where there isn't an existing paper to replicate to begin with, if it works, you're great. If it doesn't work, then it's a little more challenging to figure out what went wrong. But it sounds like, do people remember who said what? Because there were, so you, I guess there were 3 people who were talking about epidemiology models, 2 people talking about cancer models, 1 person talking about interest, immune response and viral infection in the individual, 1 person talking about neural networks. So I think it's a good range of problems. So I think the thing then is to keep working on this. Really, there'll be some homework problems this weekend, but I would encourage you really to try to develop these ideas more over the weekend. And by the middle of next week, let's have tried. I'd like to talk to everybody one-on-one so that we can, doesn't have to be completely final, but at least nail it down so that we have an initial deliverable for everyone. Get things done that way. Josh, do you have any suggestions or comments about this?
Yeah. So if people want to talk, feel free to reach out. If need directions in terms of how to conceive your projects, email. We can get back to you as soon as we can. We'll get back to you. And then the other thing is there's a couple of comments about developing GUI tools. I personally haven't tried them either as standalones or wrapped around Tellurium, but that is something that I would be very interested in working on. So if people that's, if people want to work it together, I'll be more than happy to do so as well. I'm not sure. I'll have to talk to Andy to see how to integrate some of these GUI tools with the Jupyter notebooks, but we can work that out together.
Sliders are really easy in Jupyter Notebooks.
Right, sliders are easy, but if people want to develop more like window displays and stuff that are more standalones, that people don't have to interact with the underlying code,
Right.
although that goes a little bit beyond the scope of the class, it's also something I am interested in. I would be willing to work with people.
I think you can you can hide the cell so that the cell will display its results and have an interface, but doesn't actually show the code inside of it. Gilberto's been working on this a bit, so he might be willing to help out too. He was working on getting the Glazier paper up as a example. So good, but I think people are making good progress. The key is to keep moving forward on it, and I'm pleased everyone was able to get so much done. So for people who don't have papers picked out, please try to identify at least a couple of papers. My request for today was there a paper on sort of epidemiological modeling that people thought would be really good to work on together next week? If there is, that would be fine. If there isn't, that's fine too. We don't have to spend that much more time on epidemiological modeling. But if somebody had a suggestion now, I know some people had sent some things in last week, but if you had something this week that you wanted to bring up the attention of the class, that would be fine.
That paper, the first one I cited in my presentation, it was pretty interesting as it used that cosine function, which might be interesting to look at in class. It also included a vaccine integration, which we might want to look at. And I think it was an SIRS model, which seems kind of like the next logical jump for us.
Can you share the title in the chat?
Or do you have it? Do you have the PDF of it? You could maybe screen share just the first page of the paper.
Yes, I do. Let me pull it up and I can screen share it. Okay.
Yeah, don't want to put you on the spot, but if you had it.
No, you're good.
Valencia. Okay, so it's an age structured model. Okay. Can we scroll down a little bit? All right. Let's see.
So the final step they take in this is basically a cost analysis of the hospitalizations of these children that are patients. But the modeling of the infection and the hospitalizations really is kind of interesting. Here's their kind of set of equations, and they're also pretty clear with all the parameters.
Okay, so so they just have a juvenile and an adult.
Yeah, I think the first cohort is individuals less than 1 year of age, and then the second cohort is everybody else.
Okay, good. I get nervous when I see age-structured populations because they're actually a pain to deal with numerically. If you do continuous age distribution, if you look in this textbook by Brower, he's got some age structured models, and they can get really messy. And conceptually, they're not bad, but actually solving them numerically winds up being a bear. If it's a compartmental model where you have young and old, then it's not bad. If you have a continuous distribution of ages and you have to do an integral differential equation, it's a bit of a pain. But this actually looks, I mean, this looks mathematically and numerically tractable. Now that's said without my sitting down and trying to work through it and see how it is. But at least in principle, structurally, this doesn't look like it's too bad. So I think it's possible. We'll definitely remind us, send us the link again as an email. And why don't you send the PDF around to everybody in the class? You've got the class email that I've been using. Just do a reply all to it and send it around.
Okay.
And we'll take a look at it the next day or two. And if it looks practical, we could try working on that together. That would be fun. Unless if somebody has a strong vote for something else, that's okay. But let me and Josh take a look at it and make sure there are no, we can't make absolutely sure, but try to make sure that there are no minds hiding in that paper. So that doing a replication isn't too.
If we can replicate it one day, then it should be good.
Sorry, I missed what you said, Josh.
If we can replicate it in one day, then it should be good. That's the benchmark.
Yeah. So, but that means you and I have to work on it together. We have to get Amber's stuff done. I mean, the irony is that all the people, I mean, we're working with a lot of the people whose names are showing up on these papers, but they're also knocking on our door saying, why are you spending time teaching rather than working on the things you're supposed to be doing for us? Which of course is sort of unfair. Actually, they're teaching too. He's doing a lot of teaching. Bard is teaching a lot. Amber Smith, whose paper we heard about earlier, she's in a medical school, so she doesn't teach. So it's one of the big advantages of being in a medical school as a faculty member. The disadvantage of being in medical school as a faculty member is you're generally not paid. You have to pay yourself. So if you don't have grants, you don't get paid. And anyway, teaching is an enjoyable thing. So I didn't mean to make it sound like a minus.
Just by looking at equations, it doesn't seem to be extremely complicated. And the fitting algorithm that they used in the paper is the one that we're going to be showing. I mean, the method is the one that we're going to be showing.
Great. So. Okay, so that sounds like a great introduction to going on with the class, huh? Okay, great. Well, thank you, everyone. I appreciate all the things you've been trying out. And let me come back to the lecture. If I can find it. Where we go? Oops, no. Screen share. Okay, so I'd like to talk more about parameter estimation. And we'll do a little bit together. Josh has written two nice notebooks for that. And then we will go back to do a little bit of stochasticity, and I hope begin to do some of the other model classes that we heard about today. I don't think we'll get through all of them today, but we'll see how far we get. So we saw last time that the base SI model was not a very good model for the disease. We know people recover, so by definition, SI wasn't a good place to start. And we wrote down this idea of an SIR model that you're susceptible, infected, and infectious, recovered. And the idea with an SIR model is once you're recovered, you're either dead or immune. And we've heard also that that's not a very good model for SARS, probably, because people who are recovered probably become susceptible again. And so SIRS models are probably more realistic. And we may get to SIRS models either today or next week. But the basic idea with an SIR model was you have to have some model of the rate of new infections. And that model we wrote in the form of N, number of encounters per individual per day, times gamma, the probability that when you encounter an infected person, you get infected, times the fraction of people who are infected, which is the number of infecteds I divided by the total population S0. And then the net infection rate, that's the rate per person. The net infection rate has to be multiplied by S, which is the total number of people who can get infected. We're going to assume to begin with that you have exponential recovery, which is clearly not accurate, but gives you a typical timescale for recovery, 1 over mu in days. And that's the standard structure for an SIR model. Different people name the parameters differently. In particular, N times gamma, which I've broken out here separately, is usually called beta. And beta divided by mu is R0. And so we looked at that. And the very first thing that I want to do is come back to what we did last time. And I realized after class last time that I had been making a elementary mistake in what we did. Ironically, that elementary mistake didn't change our results, which is a bit funny. And so we were looking last time at data on infections from the Johns Hopkins site or Worldometer. And one of the things that you see there is the typical headline numbers that you're given are the number of new infections per day and the death rates per day. And what we did was we looked at the number of new infections per day and tried to infer some of the parameters for an SIR model from it. And I want you to just take a minute, 2 minutes to think about what those data sets are. You could definitely go to those sites. Why don't you actually take a second or go to the Hopkins site or Worldometer site and see what the data sets are that are available. One of the data sets you'll find is total number of cases. One of the data sets will be number of new infections reported per day. One of them will be total deaths. One of them will be number of new deaths per day. And I want you to think a little bit about what data sets would be good for estimating. We really only have in this model a couple of parameters. We have mu, the recovery rate, which is 1 over the time you're sick. We have R0, which is the typical number of people an infected person infects during the time they're sick, or comparably N gamma times S over S0, which is, or beta, N gamma, which is beta, which is the number of people an infected person infects per day. And then S0, which is the total population. And the total population might seem to be something that is unambiguous, but it's not so simple. And the reason it's not so simple is that not everybody in the population is exposed, right? We've talked about the fact that somebody could live in a big city, somebody could be in a rural community. And so not everybody is susceptible to being infected at the same time, unless maybe you're on a cruise ship or an airplane where the mixing is very effective. The effective population that you're talking about is much smaller than the actual population. Another factor that one has to take into account, especially for a disease like SARS-CoV-2, is that many people aren't tested and are never diagnosed. So many people may have the disease and not know it. And so those headline numbers represent glimpses of information, but not complete information by any means. But the elementary mistake I made was a different one. And I want you to think about for a minute, if I want to estimate the recovery rate, mu, and the infection rate, R naught, or beta, if you prefer, what time series parameters would be the most useful for it? So I'd like you to look at those data sets, see which ones are available, and then take just a few minutes, maybe 2 or 3 minutes to look at that and think about it. And in particular, you might ask the question, are there any of the data sets that are out there that you can't use to estimate the things that we want to find out? So this is sort of a thought exercise more than anything else, but let me give you just 2 minutes or 3 minutes to think about that question. Does anybody have any ideas they want to share about that? One thing I could ask is, do you remember what data set we looked at last time? What we looked at was the rate of new infections. And we looked at the rate of new infections, and let's see if I have it here. Here's the rate of new infections, and the rate of new infections comes up, has a peak, and then comes down again. And then we compared that to the rate of new infections in SI model, which is okay. But if we jump to the SIR model, the rate of new infections, and there's a typo here, that N shouldn't be there, ds by dt is the rate of new infections. That's N gamma I over S naught times S. And particularly and prominently absent from that rate of new infections is the rate of recovery, mu. And if you have beta here and you want to get R naught, you need mu. So the rate of new infections cannot tell you either what the recovery time is and rate is, or what R naught is. And I merrily went in and said, okay, we're going to assume that the tail of that distribution goes like di by dt is minus mu I. And the leading edge of that infection looks like this, and then we were going to use that estimate. And it happened that when we did the fits, we got a number that was reasonable. But it doesn't make any sense at all. And now, the other data set that you have is the number of active cases. Number of active cases is supposed to be I, and that reflects both the number of new cases and the number leaving. The problem with estimating that is that, one, you don't really know how the number of active cases is counted. It's probably the number of people who are hospitalized, which may or may not be a good proxy for what's really happening. And two, it's a little bit hard to understand how to pull out the recovery rate. Now, in the case of Italy, where they locked down, so we have an idea that as of day X, the number of new infections was pretty small, then that shape of the curve that we just had isn't a bad estimator. But if you're in the United States where they never locked down, and therefore you have continual new infections, it's very hard to get that recovery rate that way. Now, of course, in the case of the recovery rate, it's not necessary, particularly, that you estimate it from the population. If you're sick and you report to your doctor that you were sick for 3 weeks, we know what the recovery rate was. And so there can be clinical data on individuals that allows you to know the recovery rate without having to look at the population. One thing that you find a lot with ODE models is that there are parameters like the recovery rate that are actually very hard to get out of population data, that are quite easy to get out of individual data. When one does cancer modeling, you run into that in spades. The size of a tumor is remarkably uninformative about what the cells inside the tumor are doing. Because if the tumor is growing, it means there's cell division going on, but you could have a very rapid cell division and a lot of cell death, or very slow cell division and no cell death. And the net rate of growth, which would be the difference between births and deaths, would be the same. You could have a few cells that divide a lot and most cells not divide at all, or every cell divides slowly. And so all of that variation between individuals and between births and deaths is hidden when you look at an aggregate measure like the total number of infected individuals. In that sense, the new infections is nice because it doesn't, and then in the argument for new infections, I said we were going to neglect mu. The argument for new infections, if we're measuring new infections, there's no mu in it at all. It really is DIDT, the forward part of DIDT. So I also did that wrong. And so actually we looked at a data set that by definition couldn't give us the numbers we want. So I'd like to come back now and try to do this right. And I'll turn it over to Josh. I'll screen share it to Josh to do these because he's the one who wrote the code. We will go back and now we'll look at the number of active cases and we'll redo the fits to get the estimated numbers. And I should probably have created something like a Google document where people could put their estimates in. But I think when we do this, probably we'll all get the same estimates if we pick the same ranges. And so maybe for today, we're all right. We should keep track of we have to keep track of these numbers. So somebody has to write down these numbers that we get because we're going to estimate first mu, then R naught, and then S naught. And we're going to need those numbers later for the next exercise. So I'm commissioning somebody to keep track of them. Somebody willing to volunteer to keep track of them? I'm hearing stunning. Thank you very much. Okay, thank you, Cal. Okay, so Josh, shall I turn the screen share over to you? And
Sure.
People should follow along. The code is lecture 4 exercises estimates. And
So people can find the code. I'm sharing the Google Drive link. If you guys go into that link, we're gonna be using exercise 1. We need the data sets, and these data sets are gonna be the active cases. They're also uploaded in Canvas in case people wanna download them from there or just remember where they are. But we're gonna go ahead and log into NanoHub, and then as we've done before, we are just gonna upload the files. I'm gonna upload Italy, Spain, and New Zealand. And then I have to upload the notebook, which is lecture 4, exercise 1, estimates. So when we launch the notebook, I am importing a lot of stuff that we are not gonna use now. It's just because this was all part of the same code. The only library that we're using here is matplotlib just for plotting purposes.
and you need NumPy too.
Oh, right, and NumPy. Yep. This is just a SIR model definition. We are not going to use this now. This is for the fitting algorithm, but it's just there. And the idea, as Jamie was explaining, is that this notebook walks you through how to estimate the parameters used by getting a rough estimate of the parameter values by looking at the experimental data. And as James was explaining, the data that we have corresponds in the SIR model to the I variable, the infected individual variables. It's not entirely clear to me whether we can one-to-one map active cases to infected individuals, because of there can be multiple reasons. The main one is that we're not testing everyone, so you don't know you don't really know how many people are really infected in the population. So you have active cases here referred to the people who get tested. And then the other part is the active cases is calculated by taking the total number of cases and then subtracting the cases that recovered and the cases of people who died. It's not also clear how you quantified people who recovered, right? Because people usually get tested once and then they test positive. If they don't develop a chronic disease, they don't necessarily go to a doctor later on to get tested again. So you really don't know how long that person stayed in the infectious case. So most of the those recover are estimated from people who got hospitalized or people who got tested in multiple occasions. But for now, we're gonna use these active cases as a proxy of that IT variable in our SIR model. And in the SIR model, the change in the number of active cases, it's given by this equation, n times gamma over r s naught times s times I minus mu times I. We discussed a couple of classes ago that R naught can be defined as the total number of individuals a infected individual infects while infected, right? And that is given by this ratio. Also, and I think James did you work already over this estimate? No, you have it later on too. You have it later on as well in your slides. But we can estimate the maximum number of people that are gonna get infected, or conversely, we can estimate our S naught once we have one of these variables. We have R naught or we have I max. Since we have I max, then we can use that quantity to estimate S naught, which is a number that we don't know, right? Which is the effective susceptible population that is not to be equated with the total population. So in this cell, I am just uploading the data set and plotting it. So we can see here what the curve is. If this is the Italy, but if I change to Spain, I get a different curve. This one has a little bit of a bump here. Comes from updating the way countries are updating their values. They suddenly realize that they've had all these cases that they were not counting, and then they dump it into the number of counted. So, but you can sort of like, when estimating, there's filters you can apply that would flatten that out, or when estimating, you can assume that it's linear in some respect or averaged over this range. And then if we can also do New Zealand, and then we get the New Zealand curve, right? For this exercise, we estimated it using Italy as an example, probably because it was a clearer curve. So as James was explaining, if we look at this curve, we can sort of estimate the values of these parameters by making certain assumptions about the epidemic. The first assumption that we're gonna make is that the rate of new infections at the late stages of the epidemic is small. And it can be small for multiple reasons. It can be small because of lockdowns, mask wearing, and in that case, you are decreasing this term, right? N times gamma, or N. But it can be also because the ratio of susceptible individuals is small too. Let's say because you're so late in the epidemic that most of your susceptible population has already been infected. So that is another case where that assumption applies. So if this term is small, right, we can get rid of it and we can rewrite this equation as just a decaying term, right? So that is gonna be describe the decaying rate of this part of the curve. So if you integrate that equation and then you solve for mu, you can then define mu in terms of the slope of that curve on of the downside of the curve. This is what I'm doing here. So I integrated it and then I solved for mu, and it's just telling me that mu is the slope of the curve on the downside of the curve. So as James
on a log scale.
Right, on a log scale. That's what you were pointing out, right? So in a log scale, it should look like a straight line. If the slope doesn't look as a straight line in a log scale, then our approximation is not good within the time range that we're looking at. Here we're looking in from day 100 to day 150. But let's say this curve changed like this and then went flat here. Then you would look then at times, you know that your assumption is good between day 130 and 150. So you will have to look at the specific areas in the curve where the slope is more or less linear. And then once you have and this is what we do here, is we plot the downside of the curve. So we start at 100, where it's starting to decline, to 150, which is the end. And then we looked at values between 10 to the 6 and 10 to the 4. We plotted in a log scale, because then we can estimate the slope, right? And that slope is gonna be mu. So the way we calculate that here is that we count at time 100, what is the value? The value is 10 to the 5. And then at time 150 is 10 to the 4.1. You can see the ticks here, you can count them. And then that goes over a period or a delta t of 50 days, right? It goes from 150 to 100. from 100 to 150. So basically, I'm taking this equation and putting it here, filling it up. When I run that cell, I get a value of mu that is 0.04. And we know that 1 over mu is the time that a person spends in the infectious state. So if we take 1 over mu, our estimate is that the person remains infected around 24 days, which seems to be more or less accurate. Is there any questions up to here? Because we're gonna apply the same to the upside of the curve. So I just wanna make sure that everyone's understands this part. Any questions? Okay. So the same principle can be applied to estimate n times gamma. And here we know that from this data, you cannot independently estimate n and gamma because it's a product of them that it's gonna affect the slope. And that product is often called beta, the infectivity. If you had other data sets, you could potentially estimate these parameters independently, but not with the data set that we're using. Let's say you had the data of the number of susceptible people, then you could use that to estimate one of these parameters independently. But the idea here now is that since we are on the upside of the curve, now the assumption is gonna be that the ratio of infected individuals to susceptible that the number of infected individuals is more, or that the number of susceptible individuals is basically as is close to S0. So, or another way of saying this is that no one's gotten infected, almost no one's gotten infected yet. So you are at the very early stages of the infection. If you make that assumption, right, then these terms, this is 1, and you can rewrite this equation in this way. And then now this is describing an exponential growth that you can read when you integrate it, integrates this way. And then you can solve for n times gamma to get the value of n times gamma. And this is why James was explaining before that you need that upside down of the curve to be able to determine the infectivity, because the infectivity has this mu when you solve for it, it includes mu in it. So we applied the same principle. Now we're gonna look at the upside of the curve. If we come back here, we start looking around, we ignore a lot of this part because it's pretty much flat. So we started at 30, and then we looked all the way up to 50. You don't need to look all the way up down here. We were just looking at the beginning because we want to get that exponential growth phase that looks like this part. So we went from 30 to 50, and then we need to calculate the slope of this curve. It's not entirely linear, but we're just doing an approximation here, right? So the approximation is that when you look at time 45 and at time 35, right? So I'm looking at those 2 time points. The change in the value goes from 10 to the 4.5 to 10 to the 3.5. And that is the upside slope, right? This is this part. But to get n times gamma, I need to add mu to that slope. So beta, or n times gamma, is the upside slope plus mu. And that is the value, 0.271. James, you have the other slides explaining how to calculate this. I'll just go ahead and do it, and then you can walk people on your slides again. But now, once we have mu and n times gamma, we can estimate R naught, and R naught, it's gonna be beta over mu, or n times gamma over mu. And then once we have R naught, we can estimate S naught by also calculating the maximum of infected cases, which is around here, right? To do that, I just apply the max. And once I have the max, using this formula, I can plug in all the values and estimate the parameters. So the estimated R naught, just from this rough approximation, is 6.5. And then as S naught is around 190,000, let's say 200,000 people, which is about the size of the infection, in Italy. If you look at, if we come here and we look at Italy, yeah, 300,000 people, more or less. So we get, yeah, we're off by 100,000, but I mean, it doesn't necessarily have to be the size of the epidemic, but the fact that it's close is a good indicator that our approximations are reasonable. Is there any questions about that? So I think the 2 takeaways from this is that to estimate the parameters, we're making 2 assumptions, right? We're making the assumption that at the end of the infection, the rate of new infections is small, and that allows us to estimate mu. And then we're making the assumption that at the beginning of the infection, the slope is given, the ratio of infected to susceptible is small. So that allows us to get the upside of the curve, and then we can estimate all of the parameters by looking at the slopes. Does that make sense? Any questions?
So I think what I'd like people to do now is pull in, he's done this for Italy. Now let's change the word Italy to Spain. And you're going to have to go through, the plots and change the window of the Y limit and the X limit to get those linear regions. And then you'll have to type in by hand. It's not auto-fitted. You'll have to type in by hand in cell number 12. You'll have to get the maximum value in your window divided by the minimum value in your window, and the ending time minus the starting time for your window, and that'll give you mu. And then the same thing in cell number 17, you adjust your window so that it gives you a good view of the data set, estimate the minimum and maximum value, the start time and the end time, type those in cell 19 to max value divided by min value, end time minus start time, and that'll give you beta. Once you have beta and mu, you run cell number 9, and that'll give you R naught and S naught, and you have to look at S naught.
Yep.
And I'd like everybody to do that and come back with the numbers they get. Everyone will get slightly different numbers because they'll do slightly different windows and slightly different estimates at the top and the bottom. So I'd like everybody to do that for Spain. And let's see whether we get something that agrees with what we had for Italy, which I've written down. We won't do a formal estimate, although I guess I could create a spreadsheet and we could actually average the values people get. And then we'll take a 5-minute break. You could do it for New Zealand. And then we'll continue with the fits. How's that? So the assignment now is to Josh, do you want to scroll back and show them where to change the, maybe you can get them started. Scroll back to where you load the data set. You've got Italy, you got Spain loaded, so it's actually so you
Right,
got,
but I loaded them, so people have to load them themselves.
right. So uploading those 4, they were all from in the same directory.
Right.
And then within if you go back to your Tellurian notebook, it's in number 9 here, line 1, where you have case country equals Spain. So if you type, yeah, well, you've already switched it from Italy to Spain.
Right,
So you've made it Spain.
While you were talking,
So,
I changed.
okay, so I'd like everybody to do that. And now following through, you'll have to say, well, the downhill slope starts around time 190 or so and runs to some time 120, something like that. So you'll have to play with that a little bit to get a window that makes sense and adjust the Y scale so that you can see what the values are, and then type them in the following cell. Any questions about that? So why don't we give everybody a few minutes to try that? And then everybody can report. Why don't you use the chat to report your values for beta, S naught, R naught, and mu for Spain. And we'll take a 5-minute break and we'll come back and do it for New Zealand.
And then feel free to write down questions in the chat if you have it. If you run into problems, or reach out over here.
I mean, you can also unmute. It's not that you have to stay quiet, but but the chat's nice because at least for typing the numerical values, and it's logged. So we have them all available. I guess, again, we could create a Google Doc or something where we could put those. I can do that if you want. You might get different values for Italy too if you refit it. So I don't know if it worked or not. I created a document and shared it with everybody. Or you should be able to type your numbers once you have them. So is it clear? Is it clear what the little assignment is? Okay, so somebody, okay. So Brian, are those for Spain, those numbers?
Yeah.
Have to find.
They should be for Spain.
And Tyler, those were also for Spain.
Yes.
And if I put the numbers in wrong, please fix them. And I'm just truncating them so that they're a little easier to see. We'll need these in a few minutes, so that's why I wanted to make sure we have them. Was everybody able to get to this document that I created, this Google Doc I just threw up? If there's a problem with it, let me know. Josh, I guess I could add a line and you could put your numbers in too.
Okay, I'm just doing it right now in case people also need help. Okay, my numbers are really off.
Wow, I didn't do very well.
Yeah, me neither.
Did you put your numbers in, Josh,
I put them in the chat. I'll put them.
in the table? Is everybody sure they loaded the Spain data set?
Fairly certain.
Okay, I believe you. I just everybody else got nice consistent numbers, and Josh and I got terrible numbers. So I'm wondering what we did wrong, what you guys did right that we did wrong. It will depend on what time range you pick.
Right.
And so maybe you picked a better time range than I did. And we'll see in a few minutes that these numbers are very much a rough out, and but we will need them for the next thing we're going to do. So. I guess I should probably have made this a Google Sheet instead of a Google Doc, and then we could have actually done the averaging in it.
It might not be that hard to copy and paste the values over.
Yeah, you should be able to just copy it over to an Excel.
I wonder if I could turn it into a sheet.
Or sheet.
Let's see. Yeah, you can do it. Just cut and paste it, it works. So let's see.
Callie, you and I are up there in the Spain data set.
Yeah, I don't know why my beta is so different from everyone else's.
Whoops.
I don't know.
Well, we're eyeballing it, so it's not.
Right, look at the R knots. We got, most of them are around 6, but James and I at the high end. And then Antonio and Callie are on the low end. But it's all gonna depend on what portion of the window you're using and how good, or I mean, how you're eyeballing your approximation. Mine is pretty rough. I was like counting like quickly and not very carefully. But we will see that we're not gonna make policy based on this. We'll need it later on. So, James, should we?
So I think what we should do is we should take a short break.
Right. I need a break.
And then when we get back, I think instead of, I mean, if people want to fill in the missing pieces, that's great. And but if we don't, I think we'll continue. You can do it as a little homework exercise.
Right.
And because I want to go on to the fits,
Right.
because that's the more exciting part of this.
Right.
And we can use the rough numbers that we have from here. But it is interesting to see what kind of variation we've got. I'm not sure why Josh and I did so much worse than everybody else. But that's great. So let's take a 5-minute break and we'll come back at 7:10 and go on to doing some fits. Okay. Oops. Josh, whenever you're ready.
Yeah, I'm back. I was wondering if you wanted the screen back or should I just continue with
No,
the.
why don't you continue?
So, so far we estimated our parameters, these parameters just by looking at the regions of the data set and basically eyeballing the slope. Alternatively, we can use more sophisticated fitting algorithms that are gonna determine all of these parameters at once. For today, we're just gonna introduce general idea of fitting. We will come back later to discuss different fitting algorithms in quantification of certainty with parameters and parameter determination. But today it's just like as an introductory, just to give the flavor of fitting. So the basic idea with fitting is, let's start with the assumption that we have a data set that looks like this. We've collected data in time, and let's say this is the number of susceptible or the individuals in my population, right? So we collected this data, we have the experimental data, and we propose a model in which the change in the population is given by this form, right? That the number of susceptible individuals is decaying exponentially at a specific rate K1. When we make that assumption and we assign K1 a specific value, then our model makes a prediction about the change in population, about the population in time. And then once we have that model prediction for a specific value of K1, different values of K1 will generate different predictions. But for a specific K1, we can then measure the distance between the experimental data and the model prediction to quantify the error, or what it's called the residuals. Okay, so that is important because that is what our algorithm is gonna do. Is what our fitting algorithm is gonna do is to test different values of K1 such that the distance between the model prediction and the observed experimental data is minimized. So, as I said, the fitting algorithm works in a way that it tests or adjusts different values of the parameters to minimize the differences between our data points that we're calling here X1, Y1, and our model prediction. So the distance would be the absolute value of the observed data point minus the model prediction. And there is multiple ways in which we can quantify the whole error of the model. That quantification of the whole error, it's called the objective function. And one of the most common objective functions is the sum squared of the differences, what's called the chi-square, where the idea here is basically that you iterate through every observed data point. You measure the difference between the model prediction and the data point, and then you square that difference. So it doesn't matter if the model is over predicting or under predicting. And then you do that for every observed data point. And that is gonna be chi-square. And that is hear me? James, do you hear me? I'm
I
sorry.
hear you.
Back, you dropped out for a
My,
second.
my AirPods are connected.
There. No,
So,
there. That's okay.
yeah. So, right. So the objective function, the quantity that the fitting algorithm is going to try to minimize is this function. You can have multiple different functions. You can also have very complicated functions. So we were talking to a friend of ours. Sorry. A friend of ours was also working on fitting some vital load curves or vital data. And she, instead of caring about every single point in the observation, there was a particular point that was more important for her than all the other points. So you can weight the points differently such that one point is more important than others. In this case, we are assuming that every point is equally important. But depending what's gonna determine the form of this function, it's gonna be specific to the problem that you want to solve. So the way the optimization algorithm works is that you start with an initial guess of those parameters, and then it computes that objective function, the value of that objective function, and then adjust the parameter values such that the objective function is reduced in the next iteration. And then it keeps repeating this process until the function cannot be reduced further. This function is going to be it's a function of the parameters, right? So it has a dimensionality that is equal to the number of parameters. In the case that you have, let's say, 2 parameters, then you can visualize that function as a surface. And what your optimization algorithm is going to do is that let's say you're going to start at a point in that surface, right? At a combination of 2 parameter values, 2 sets of parameter values, and then it's going to move in that surface to find a point where the surface is at a minimum. The surface can have multiple minimum values, and there is different algorithms that are designed to address some of these issues, because what you want to find ideally is the minima that is the smallest of all the minimas in your surface. So that's called the global minimum. But depending on your algorithm, you might get stuck in one of these, what it's called local minimas. And then there's, we'll come back to this to discuss how different algorithms deal with getting out of these local minimas. For now, the idea is that our algorithm is gonna move, right? So this is basically the value of the error between the model and the data. And our algorithm is gonna move such that the error is minimized. So it's basically driving me from here to this point, to the global minimum, okay? Let's see. Is there any questions about that? How many of you have have heard about parameter optimization before? Has anyone heard about it before?
I don't think I have.
Okay, cool. So this should sound familiar. It's very basic for those of you who have heard about it, but for those who haven't, we'll come back to it in more detail later. We just want to show now how to this can be used to apply to estimate the parameters in a way that is not just eyeballing it the way we did it. So if we go back to our notebooks, in that same folder, I have uploaded a second notebook that it's called Lecture 4 Exercise 3 Fitting. When we upload that one, we open it. The first part of the notebook is just gonna be what we've done, what we did so far to estimate the values of the parameters. Because we're gonna need those estimates. If we come back to how the algorithm works, we need an initial guess of parameters. So we're gonna use our estimates as our starting point to compute for the algorithm. So that's why James was mentioning that it's important for us to have these numbers, because we're gonna use them as our initial guesses. So basically we're choosing a point on this surface to start with. Here, we'll come back to this, but what I've done here is I have defined my cost function or my objective function such that the load is model every time this function is called. Then the function takes as an input values for the different parameters of the model. So what I'm doing the next lines is I assign to each parameter of the model, the values are being passed on to the function. Then I simulate the model, right? And then once I've simulated the model, I go over each point in my data set, in my experimental data set, and I compare it to the model prediction, right? I subtract, I get the difference and I square them, right? Which is basically I'm applying this function, right? I'm going over each data set in my experimental data. Line 11, what it's making, and we can discuss this later, but I am making sure that I'm comparing points that are the prediction, the model prediction and the observed data are occurring at the same time or close enough, because they might or might not be equally spaced. And we can come back to that later. But the idea here is that in line 12, I am counting the difference between my experimental data and my model prediction. And then this function returns that difference, the whole error between the model and the experimental data. We're gonna ignore this for now. We'll come back to it later. We're using to wrap our fitting code, we're using this library that is readily available. I'm gonna post it in the chat. We're using this library that allows us to compactly define what are the parameters of the model that we want to fit, and then allows us to define what are the ranges in which these parameters are going to be explored and what are the initial guesses of these parameters. So we can see that the initial value that I'm passing for S1 in my model, I uploaded up here, right? And it has the parameters beta, K1, and then it has S0, which is called here S1 at the beginning, right? So, and it has also I1, which is the initial, it has the initial number of susceptible, the initial number of infected, it has K1, and then it has beta. K1 corresponds to mu, and beta corresponds to beta times S naught. So that's why we here as an initial guess for S1, we pass our estimated S naught. This one is not that important. We'll just set it to 1. For beta, we pass on our estimate of beta divided by S naught. And then for K1, our estimate of mu. And if we run that, I'm sorry. If we run that, we can see, okay, I have to run everything. Okay, so if we run that with our estimated parameters, we can see what our model predicts compared to our data. So what do you guys see here? What is this telling us about our estimates?
Are they good or bad estimates?
It's off kilter, isn't it?
It's kind of off. It's close, right? But it's still quite off.
Yeah.
So it's a good start, right? And that's actually what we need. We need a good initial guess. Because now it means if we look at this surface, it means that we started somewhere around here. We could have started out here with a very crazy number of parameters. But the fact that we're close, it means that we're someone around here, right? And then what we're gonna do now is that we're gonna let the algorithm take us from here to the minimum. And then when we run this, and then so the paper that I think was it Nico? The paper that was suggested earlier uses actually, when I said the method that they use for optimization is actually the same method that we are using here. So what we're gonna do now is we're gonna let our algorithm try to find the parameters that minimize that difference. And you guys can see how the algorithm is changing those parameter values to try to get a curve that fits better the data that we provided. And you guys see how it's exploring that space. It's gonna take a little bit for it to find it, but keeps going and going and going. And you guys see, guys can see that by now it's getting really close. It's pretty close right there. We could have, if we want to be fancy, we can actually tell the algorithm when to stop, when we say, okay, this is good enough. We're not doing that now. We're just letting the algorithm decide when it's good. But you guys can see that it's quite close now. So this is when where we started with our initial guesses, and this is where the algorithm is taking us. But it's not changing a lot in between these steps. So we could, yeah, if we had a sophisticated code, we could tell the algorithm, it's not, you haven't changed for the last number of steps, so means this is quite a good approximation. But it's eventually gonna stop it's gonna stop at some point. Right. So once it finishes, now we can compare what the model was, what the model prediction was with our initial guesses of the parameter, and what our model prediction is after running the fitting algorithm. Okay. And then the fitting algorithm is going to report what the optimized parameter values are. And you guys can see that in terms of the parameter of these parameters, S0, beta, K1, we were not terribly off. We were more off with our initial guesses of I1, which is not something we estimated from the data. And then when we print them out, the results, right. So now we get an estimated R0 that we can confidently talk about in terms of reproducing the data. And this, in this case, is 3.68. And the duration of the infection is 26 days. And also the effective susceptible population set out 297,000. Look at the cases from Italy. So we're pretty close. Okay.
So Josh, why don't you put those numbers into the table in that Google spreadsheet that I just did?
Right.
And so why don't people, did everybody follow that? Why don't you try doing it for Spain?
Is there any questions? In terms of changing the code, there's nothing you guys have to do at this time. We will come back to it later, and then we'll let you play with what data to adjust, what parameters to adjust, how to estimate the ranges and stuff. But for now, we're not doing any of that. We're just using our estimates and let the optimizer run.
Have people, I guess you asked before, but have people done fitting before? Yeah, I guess a show of hands if you have, or if you've done this kind of fitting before, maybe put a wave or something in your screen box so we can just, because it's something that's critical to do. It's not trivial to do. Okay, so Kali's done it. Okay, Tyler's done it. So some of you have done it before. In which case I beg your patience for those of them who haven't. It's as you know, for those of you who've done it, there's a certain art to it. It's not so easy to get it to work. In particular, if you start with estimates that are way off, or the bounding ranges you pick aren't appropriate, very often you'll get your algorithm will run off and happily grind away for hours, days, weeks, however long you let it sit, and give you nothing. And so the reason we went through this exercise, which really took the whole class, is because it's critical to be able to build a rough estimate using something that's easy, like measuring the slope of the line with a ruler, which is more or less what we did first half of the class. And then use those estimates to then do a more sophisticated fitting algorithm. And later in the class, we'll also talk about uncertainty quantification and the likelihood estimation, which is this algorithm will give you a number, which it says is the best fit. But what you don't know from that is how certain are you that that's the number. In other words, could the number be twice as big, half as big, 10% bigger, 20% bigger? And in principle, it's possible that they're trade-offs, so that if you made one of your parameters twice as big and one of them half as big, you wouldn't change anything. And again, some of that's a bit of an art. Some of that can be done in a more systematic way. One thing that I find a bit surprising about this is that we know that the SIR model isn't a very good model of what COVID is doing. Right? We know that the reality is more complicated than an SIR model. And so I actually expected the fit to be pretty bad when we started doing this a week or two ago. And I was rather surprised that the fit actually is pretty good. And so if you ask the question, if we need a very fancy, do we need a very fancy model of this virus, the epidemiology of this virus? Well, we could write down a fancy model with SEIR, quarantine, et cetera, et cetera, et cetera. But if the SIR model by itself already fits the data very well, how could we tell that we need the fancier model? And so that's something to think about. I'm not going to give you an answer to that.
Right. I do want to say that we've cut off this data, right?
Mm-hmm.
So the active cases in Italy look really like this. But the SIR model is not going to give you this upside curve. So that's why we cut it here.
I mean, the other thing is that we've assumed that nothing changed during the course of this infection. Right. Whereas we know that Italy locked down, which is why there's the peak.
Right. And
so
that paper...
so it's what's called a misfit. In other words, our model really shouldn't fit the data, except that if you assume, what would this correspond to? This would correspond to the idea that there were 300,000 people in Italy who were potentially exposed to the virus. And then there was a perfect lockdown where nobody else was exposed after that. That would be a reality that could give rise to the data set that we see. Of course, it's not as simple as that. The places where you might get something that looks like this would be, say, a cruise ship, where you had people locked together for some number of days, 4,000 of them. And in that case, you might actually have something that looked more like a classic SIR model. But it's actually, to me, rather interesting that this simple SIR model, which clearly doesn't describe the reality of the infection very well, actually reproduces the time series quite well. So any questions? Why don't we spend a few minutes trying to do it for Spain? Let's see what happens. Have you done it for Spain, Josh, already? Let's try. Let's all do it together. See what we got. I haven't done it, so give it a shot. If you go to that Google spreadsheet, I've got the averaged values that people guessed. You can use your own guesses, the ones you came up with. You may have to adjust the bounding ranges. So
right.
People have
what
trouble
I'm going to do is, since I'm not going to go over all of that again, I'm just going to define them up here. So we have beta. Well, I can define them down here. Can someone tell me what the average estimates were for the parameters for Spain?
0.271 was the average beta for Spain.
0.271.
Mhm.
What about you?
0.0399.
And then R naught.
7.34.
And then S naught.
189,225.
235.
89,200. Yeah.
Okay. So that's our. It's not. It's actually not that bad. Right. So this is the Spain data from Spain, and then this is the model predictions that we got from our averages. As I was mentioning, you can have, because these are kind of like outliers, you could have complicated cost functions that weight the data depending on how far away they are from, let's say, an average or whatever, whether there are outliers or not. We can come back to that and think about it more. We can think about it later, but for now we'll just try to fit it to this and see. We haven't done it, so let's run it.
It actually works surprisingly well, given that big glitch at the peak of the Spanish data.
Right. But I'm interested to see what it's gonna do about it. What it's gonna, is it gonna ignore it, or is it gonna? It's probably gonna average it. Yeah. So something important, and I wanna, James mentioned it already, but I wanna mention it again, is that the good of your fit is gonna depend a lot on the goodness of your initial guesses. We spent 2 weeks trying to get a fitting algorithm that worked when used, and it's exceedingly complicated when you don't have initial guesses, because your algorithm can just fly off. You can always start moving around this. If let's say all of this is flat and you start out here, it's just gonna move on the flat areas, which is what we were seeing. It will never arrive to a good local minimum or a global minimum. So that's why we spend some time estimating, eyeballing those parameters, so we can have an initial guesses of those values. So this one is kind of weird. The duration of the infection is very long now. It's 45 days, and the R naught is bigger than the one we estimate.
Really? Oh, sorry.
Beta is about the same. K1 is not terribly different.
Right, I think it's getting the R is because it's assuming it's coming up with the duration of infection being very long.
Right.
Which means mu is small, and since our beta is turned into R naught by dividing by mu,
right.
If your recovery time is off,
right,
then you'll get the R naught that's too big.
but it's actually fitting the data quite well.
Right. So that suggests there's something about reporting on, especially on the recovery side.
Right.
It's it looks like the incidence data are probably pretty good, but the reporting data are not.
Right. But these are interesting questions, right? Because the model, the parameters do match pretty well the observed data. So you want to ask yourself, why is it that this number doesn't match our intuitions of what the infection, how the infection progresses? And then you can ask certain questions about the data itself, not necessarily the model.
And these values that don't match our expectations, is this an indication that we're using the wrong type of model?
That's certainly one possibility. Another, of course, is that the data sets themselves are corrupt in some way. As we mentioned, exactly how they take people out of the active cases category is complicated. But yes, I would say that if you fit all the data, if you take the countries that did really successful elimination, like Japan, Taiwan, Spain, New Zealand, and so on, you would expect that at least different countries might be different because people socialize differently and mix differently. But you wouldn't expect R not to be 10 times different. You might expect it to be 1.5 times different. And the recovery time from the disease should be the same pretty much everywhere. And so if those numbers are way off and you're still fitting your data, then, if I took a 10th order spline, I could fit anything with any simple curve with a 10th order spline. It doesn't tell me anything about the mechanism by which that curve happened. And so it's certainly possible that if you're seeing that the parameter that you're getting the fits, but the parameter values aren't lining up, that's telling you that somehow you're misfitting the data. And so in this case, I don't think we're totally crazy. In other words, the estimated value of R is between 3 and 6, so that's not way off. The R naught may have been higher in Spain initially. The recovery time really does seem to be strange, but we're not fitting very far into the tail, right? The Spanish data here cut off at 120. So it's possible that there are other things going on. But it does show that you could get something at least not crazy out of the basic data sets using a very simple model. And I think there's some value to that. We're almost out of time, so instead of skipping through the things we skipped through before, Josh, if you'll let me screen share, I just want to talk for a minute. I'm going to jump ahead a little bit. And actually, I don't really have the slide that I was hoping for, but anyway, we'll do it from here. Which is, we heard the term herd immunity before. So if I look at my growth rate for the number of infected, it looks like n gamma I over S naught times S minus mu I. And so the number of people who are infected starts decreasing when the argument, the right-hand side of that term, is negative. And if I rearrange this term, the I comes out, so I have n gamma times S over S naught minus mu is negative. And if I put R naught equals n gamma over mu, I get R naught times mu in this first term. And so I get R naught times mu divided by S naught minus mu is negative. And I rearrange, and that's equivalent to saying that 1 over R naught is bigger than S over S naught, or vice versa. R naught is bigger than the fraction of people susceptible. So if R naught is 8, the epidemic will stop when 1/8 of the population is susceptible. Actually, it's not quite that, but it'll start dying out when 1/8 of the population is susceptible. And that's what's called herd immunity. If I have a disease with an R naught of 2 and half the population are immune, then the disease won't spread. And that can happen in 2 ways. I could vaccinate people and have half the people immune. Half the people who get the disease and recover from it, and that's classic herd immunity. Or potentially they could have been exposed to it in the past and been immune for some other reason. What you'll see, though, is that the fraction susceptible has to be less than 1 over R naught. And so if R naught is big, if R naught is 10, 90% of the people have to be immune to stop the epidemic. If R naught's 2, half of the people have to be immune to stop the epidemic. And so that's the definition of herd immunity. There are different ways you could deal with the disease. You could reduce the number of encounters everyone has. This is part of reducing beta. That would be isolation, lockdowns. You could reduce gamma, the probability that when you meet an infected person, you get infected. That would be like wearing a mask or staying 6 feet apart. You could reduce the time over which a person is infectious. Antivirals might do that. Isolation after infection might do that. Or you can do vaccination, where you reduce the fraction of the population that's susceptible. And we could look at, at some of these numbers. I think we did a little bit of this last time. Influenza 3 to 4, that means if you don't want flu to spread, you need 60 to 75% of the population to be immune. No, we're nowhere near that in terms of flu vaccine. And if the vaccine isn't perfectly effective, then you have a problem. If the vaccine is 50% effective, that means that even if you vaccinate 100% of the population, you're still going to have an epidemic. It'll be smaller, but it's not going to get rid of it. If you look at a disease like measles with an R naught of 20, if you don't have 95% of the population vaccinated or previously exposed, the disease will spread. And so you need to have very, very high vaccination rates and very high efficacies for the vaccine for that to work. With the estimates that we're getting of R naught of about 5, we'd need to have 80% of the population immune to get herd immunity. And that means either 80% of the population has to get the disease, or you have to have a vaccine that's effective and you have to have 80% of the population vaccinated, or some mixture of the two. If you assume that you have herd immunity at 80% and you don't have a vaccine and the death rate is 1%, then that means 0.8% of your population will die. If it seems that it's a little bit lower, it's more like 0.5%. So you'd expect something like 0.4% of your population to die if you went through to try to get herd immunity. Sweden is the only country that's tried to do that officially. Their death rates seem to be a bit lower than that, fortunately. But it's not easy to get there. And even Sweden doesn't seem to be there in terms of the number of people infected. The other problem, of course, is that long-term consequences of the disease. Even if you don't die from it, it can leave you disabled. So that's a nasty consequence. But you can get these numbers. So here's R naught. Here's the fraction 1 minus 1 over R naught that you need to vaccinate successfully. And you'll notice that there's a lot of uncertainty. Look at malaria. The estimates of R naught are 4 to 30. So that's a little bit difficult to work with. Flu, here they've got number of 2, here they've got number of 4. So there's some big uncertainties of these. So it means, again, that if you want to use vaccination to stop the disease, you have to have the vaccine has to be highly effective, and you have to get a big fraction. Because if you're off, you're still going to have spread. And so, again, that's one of the reasons that people are so obsessed with R naught. Notice that we're getting estimates of R naught of 4, 6, 8. If we want the disease to go away, we have to get R naught below 1. And that means that you have to change whatever it is you're going to do, whether you're doing it by isolation, quarantine, social distancing, vaccination. You have to get R naught down a lot. Because if R naught's above 1, it will spread. And so, in a sense, knowing your enemy by knowing R naught tells you something about how hard it's going to be to suppress the disease. Again, we have influenza epidemics every year despite having a vaccine, and lots of people die from flu every year. Now, some strains of, it's certainly not true that SARS-CoV-2 is just another flu. In general, SARS-CoV-2 is pretty lethal, and it spreads very easily. However, there are strains of flu that have been present in the past 50 years that are more lethal if you get them than SARS is. Fortunately, those strains of flu have tended to be less contagious. So if you look at this influenza, 1918 influenza, the terrible pandemic influenza that was so lethal, the estimated R naught is 2. And so it was actually less contagious, even though it killed a larger fraction of people who got it. Classic SARS, SARS, had a lethality of on the order of 10%, but it also had a lower R naught. And so all of these kinds of models assume everything stays the same throughout the course of the epidemic, which of course it doesn't do. Even if there's no government intervention, when people start dying in the streets, people hide. And so at that end, the number of people you encounter goes down, even if there's no policy decision to do that. So those are some of the things one can think about. So I'm sorry that we didn't get a little bit further, perhaps. But next time, I promise you we'll do stochastic models. We'll talk about some of these other SCIR models. We'll take a look at this paper that was suggested. I don't know if we'll get to it. We'll try to get to it next time. I hope to talk to people about projects. And we'll try to write up in the next day or two, we'll try to write up a little homework assignment. I think you can assume that doing the New Zealand fits will be part of your homework assignment. So if you want to get a jump on it, you can go through exactly what we did already for New Zealand. I'd also ask you, as a part of the homework, to find one other data set. We've done Italy, Spain, New Zealand. Find a data set for another country and do it. See what you can come up with. Okay. Any questions before we break? No, it's late. Thank you again. I'll see you next week.